So we are here to discuss about the panel discussion about the abstracts which are presented at WCLC and ESMO. And the first abstract over here is ipilimumab plus nivolumab and chemoradiotherapy followed by surgery in patients with resectable and borderline resectable lung cancer, the INCREASE trial. And my first reaction when I actually saw this abstract, the name of this is the first thing which comes to mind about INCREASE is the request letter to the boss for increase in salary. I mean, that's the only letter that we will write for increase. That nothing else we will write. My boss's reaction on reading this reaction is basically, no worries, you will get your salary increase when you will get more experience. So maybe that basically means that you will have less hair on your head. And now I understand why Dr. Somani has less hair on his head. So Dr. Somani, can I also see you on the screen over here with less hair? So maybe that is why the money is directly proportional to the less amount of, or inversely proportional to that, sir. And this is what my boss's reaction finally is when I go for a salary hike. And I'm sure everybody, Dr. Aditi is also smiling very nicely onto that. This is exactly what we all feel when we go to our bosses to increase the salary. But let's come down to what we actually are here to discuss about. And so what do you think? Ankur, I'll start with you first. What do you think was the reason behind naming the trial as INCREASE? Look at this. I का ipilimumab, N का nivolumab, chemoradiotherapy, R, E पता नहीं कहाँ से वो therapy में से E ले आए, S ले आए, मतलब surgery का E ले आए. This E sounds very, this thing. So what do you think was the reason behind naming the trial as an INCREASE trial?
I think, sir, maybe just showing the increase in the PET CR or CR rates.
Increase in the pathological CR. We are trying to convert a lung cancer into a breast cancer or a colorectal cancer now. Dr. Tarachan?
Ankur is saying rightly that now the more and more focus of the pharmas, you can say that company, is to increase the PET CR. They have got the surrogate marker of overall survival, that is the PET CR. So what will we lose or what will we get after giving so many medicines? Now doublet, single immunotherapy, doublet immunotherapy, and then adding something also, and then how much PET CR will give us and will take us from us. Will take
so maybe increase in PCR. Dr. Aditi, madam, what about you? What do you think INCREASE stands for? Definitely not the salary for all of us. So what else does it stand for?
So it looks like the number of drugs that doctor has to handle and the patient has to handle the adverse effects actually.
Immunotherapy, matlab Nivo, EP plus chemo plus radiotherapy, I mean.
Everything which is available on the plate, followed by surgery.
Followed by surgery. So I mean, kuch reh gaya to bata do bhaiya usme. Maybe the only thing which was remaining was the Baba Ramdev therapy or something of that sort, So that was which was basically causing a lot of headache. I mean, I didn't understand the first time I came and I said, okay, nivolumab, ipilimumab, you have radiotherapy concurrent, you have a surgery. It's a difficult trial actually. But let's critically analyze the trial and its rationale. So let me start with Dr. Lalit Mohan. Dr. Lalit, sir, are you a believer of neoadjuvant chemotherapy in lung cancer? I mean, we all keep on giving neoadjuvant chemotherapy, but to me, most of the times, if it is resectable, it's resectable. Otherwise, when that surgeon doesn't want to leave or let go of a patient, they say, "Tum like 2-3 cycle chemo de do, fir dekhte hain kya karoge." And that is how the people come to us. So, what is your take, sir, on neoadjuvant chemotherapy in lung cancer?
I think routinely in practice with the available cytotoxic chemotherapy, we do not subject them for neoadjuvant. But of course, the neoadjuvant sounds very good in fact. And like in breast also, we are seeing in high risk triple negative, which are sort of more of immunogenic drugs responsive treatment. And same thing applies here. So to me, this particular study looks very rewarding in the form that we may be having more PET CR and more conversion of the PET CR translating into the OS and DFS.
So you feel that the intent of neoadjuvant chemotherapy is to increase the PET CR and the MPR. That is what you actually feel and you are excited on. But sir, how many times do you give neoadjuvant chemoimmunotherapy in lung cancer right now?
Honestly, never. I have never given.
Honestly. How many times only neoadjuvant chemotherapy, sir? Sir, I've got a phone. Okay.
No chemotherapy. In fact, neoadjuvant setting, no chemotherapy as well.
No chemotherapy as such.
Commonly, we see in advanced stage. So at least I see not less than stage 3A or B in fact.
Okay, Dr. Aditi, how many times do you give neoadjuvant chemo or a chemoimmuno in lung cancer?
Sir, definitely not chemoimmunotherapy. But sometimes when the patient is not very convinced of the surgical resection, rather than the doctor, the patient wants some time to think and to assess the pros and cons of the surgery sometime. So yes, to buy the time, yes, we do give.
Dr. Tarachand, your take?
Sir, I have not given a neoadjuvant immunotherapy in a single patient till now.
Okay.
And even I did not find that patient is fit for surgery or very fit for surgery later on, and I gave the neoadjuvant. Usually patients come for the neoadjuvant chemotherapy followed by chemoradiation to us in the lung cancer.
But typically
Like
right now, I think what we are hearing from everyone is that, when in doubt, when the patient is in doubt and the surgeon is in doubt, then you give neoadjuvant chemotherapy. But Dr. Somani seems to have a different take. He says, and with the neoadjuvant immunotherapy coming up, we should we should get more into this. So let's come to neoadjuvant chemoimmuno. I don't think anybody has great experience. We hardly have some 5-6 patient experience also as a part of clinical trial. So let's start about this trial right now. I'll go to Ankur first. Ankur, your comments about the trial design. What do you think is the trial design? And okay, requesting Dr. Somani is asking, requesting everyone to ask questions to win prize. So the moderator also asks, okay, fine. Maybe I'm the one who will ask the maximum questions, sir. I'll get the prize. So Ankur, what is your comments about the trial design?
So basically, I think, sir, what they wanted to do in this trial was to see the, combining immunotherapy basically with modulation with radiotherapy, because they planned for chemoradiotherapy as a pre-op treatment, but they combined immuno because now the concept is coming that if you combine with radiotherapy and immune cells, the proliferation effect is more. So maybe that was the rationale behind combining immunotherapy, but I don't know with CRT, which is not the standard of care, like immuno plus CRT, then surgery. So
okay,
it's
Dr.
a little bit complicated, or not a trial which can go directly into practice.
Fair enough. Dr. Aditi?
So too many modalities to be used at the same time, of course. The patient has to be very fit. Sometimes we generally don't get in the clinic so fit patients of lung who can tolerate each and everything of it. Plus, a surgeon also has to be very confident enough of dealing with all these therapy side effects during the surgery.
Okay.
That the patient is.
Sir, same with Aditi, and usually so many therapies in a neoadjuvant setting. It will be I don't prefer using.
Okay. So there were two things that came to my mind. One was only 26 evaluable patients which were basically seen on this. So that was something which is very low. I think let's not criticize the RTCT a bit too much. This was basically used in patients, if you see over here, on invasion of thoracic wall, vertebra, mediastinum or diaphragm. So this was basically used in patients with pancreas tumor. So that was where you actually give chemo RT and then surgery later on. So I heard the discussion which was there in ESMO by the presenter. He said they were a referral center for only pancreas tumor. So most of the patient, and I'll come to you in the next one, these were the ones who basically had a pancreas and then they were given induction chemo radio along with the doublet immune, and that is how they underwent surgery after that. Then how is this trial different from the other trials? The one very important point again over here, these are the all neoadjuvant trial, and what Dr. Somani was and Dr. Lalit Mohan Sharma was saying, most of this trial like Checkmate 816, Empower 030, Keynote 671 and AGIAN study had resectable patient. Now this was a patient which tried to convert a borderline resectable to resectable. So in breast cancer, we have a locally advanced inoperable breast cancer to operable breast cancer. You have a locally LOBC, that is locally operable to a breast conservation. So this was probably a trial where there was a borderline inoperable lung cancer, which then got converted to, they were trying to get it converted over there to surgery. That was the difference between this trial and the other trials actually. The primary endpoint was a major pathological response. So I'll come to Dr. Lalit Mohan. Sir, does your pathologist report it as major pathological response, or they'll just write PCR or not basically? And what do you think is the ideal endpoint for immunotherapy in the adjuvant neoadjuvants? Will you look for OS, EFSJS, PCR, MPR? Do you think?
Right.
Sir? I think sir is okay. I'll come to Dr. Ankur.
So basically, they took a major pathological response because of such a small number of patients. Routinely, personally in my practice, we don't see that much of lung cancer patients which get operated, because very few patients come to us in such early settings. We usually, or I have seen only other PET-CR, not a major pathological response, which is kind of a new thing in the lung. I have not seen usually.
Yep.
As for
Okay.
the endpoint, ideally the gold standard is, ultimately OS, but I think rather than PCR, EFS is a better endpoint. But again, that needs a little bit larger number of patients for such a small trial.
So if you look, Dr. Ankur, and all the trials primary endpoint over here is either MPR, RFS, OS, and MPR. So, the world is moving towards MPR. Even the Perjeta study, the breast cancer study, was approved on the basis of increased pathological CR. So pathological CR is something which is always a issue. What I'll try to show you over here is MPR versus PCR in lung cancer. Why did MPR come into lung cancer? Was that PCR basically was seen infrequently in lung cancer, less than 10%. So major pathological response was basically proposed as surrogate marker, which was defined as 10% residual viable tumor. And then they found out that this was also predictive of long-term OS after neoadjuvant chemotherapy. So in lung cancer, you don't actually look at the PCR, you basically look at the major pathological response. And I think all our pathologists should start reporting this also very important. Now, does this PCR MMR in lung correlate with EFS? Yes, it does. This was the trial which was the nivolumab plus chemotherapy in Checkmate 816, which basically showed that it was very important. Anything to do with this, Dr. Aditi, patient disposition and characteristics, anything of interest that you would like to point it out in from this? So again, I'll ask your comments. Autoimmune disease was there in 4 by 26. I mean, 12.5% getting autoimmune disease, and Nivo and ipilimumab is in a clinical trial setting is fantastic. I mean, their ethics committee must be very commended to give so much of autoimmune disease. And the second point which came to my mind was, again, this T3, D4 was basically higher over there. And the radiation dose which was given was 50 gray. So this is basically the, this is not the definitive CTRT dose that we give 66 grays or 62 grays. It is the pre-op CTRT dose that has actually been given, which was there in the PANCOS tumor. So this was what was the important thing. Very important again over here is the induction therapy was started 26 and 24 people were operated. So a good number of people did get operated. So that's a very interesting point from our point of view, that fine, it's not that people are not getting operated actually.
Yes, sir. One more thing, can I say, sir?
Yeah, please.
Almost 60% of patients had a PDL1 more than 50% in the study.
So we will come to PDL1 also. We will come to that. Okay. This is TRAEs, which was there. 67% of patients had a grade 3 or grade 4 treatment adverse events. Now, which is also very high actually. So any comments, Dr. Lalit or Dr. Tarachand can take it. Dr. Lalit, if you are there, sir. Or maybe Dr. Tarachand can take it then.
Sir, grade 3-4 toxicities which were observed around 67% and the number 18%. I think these were all immune-related side effects or chemotherapy-related side effects, because immune-related side effects are not so observed in any trial other than also not in neoadjuvant settings.
Absolutely right. That is why I put it was not the immune-related side effects, it was actually the hematological toxicity which was there with the CTRT1. So the IO toxicity which I have put across over here are by and large similar to what we normally see. But it was the hematological toxicity which was basically seen, and you're absolutely right. Now, this is what the PCR report with Checkmate 816 is 24 and 37%, which was the difference which was there. This is the PCR rate 63% versus 79% in patients who had received induction. So this was very nice. The identified the 3 people who did not get it. PDL1 had no importance, and EGFR and all also there was discrepancy. So are you impressed with these results, Dr. Ankur and Dr. Aditi after that?
Yes, sir, definitely it's a very good result for in this setting. It's a pretty good seeing in the lung cancer setting such kind of response rates with just pre-op CRT.
Okay, Dr. Aditi.
Yes, sir. Definitely, the MPR has increased a lot. If we see from the compare with the previous studies, whether it translates into OS and PFS, that is of course a question to be asked.
We don't know. Okay, so this has basically got a 63% PCR, 79% MPR, toxicity rates were acceptable. No patient failed to undergo surgery. Do we agree? Okay, just score the trial on a scale of 0 to 10, and then we move on to the next one. Rate the trial 0 to 10. I mean, how good do you think the results are? Dr. Aditya, I'll chat with you.
6 maybe, sir.
6
I don't
maybe,
know.
fantastic. Dr. Ankur?
8, sir.
8. Dr. Tarachan?
Sir, I will stick to the 5. That is not in the favor, not the against, as the number of the patients are low, and even we can keep the finances apart, but still the finances are of matter with, and that is too great. And the PATH-CR which has been achieved, it is translating into the OS or not. It is a matter of the time also.
I also think, you know, I mean, although I am not from Rajasthan, which is the seat of the high political discussions nowadays, Delhi is always the seat of high political high command, the status which comes over there. But I also think, you know, although the PCR rates are higher, but just 26 patients, it's not a randomized control trial, good amount of people with toxicity. Basically, if your surgeon really feels, and this was not compared against only nivolumab, so now why not nivolumab plus CTRT? So it's a good hypothesis generating a trial, not something which I will take it to my practice on tomorrow or something. But yes, it's making sure that yes, neoadjuvant chemoimmunotherapy also has a role in lung cancer, and we need to define this basically further. So the next trial,
Ullas,
yes, doctor. Yes, doctor.
My camera is not fully working. In fact,
Yes,
this looks a good trial, at least in the form of, like you say, hypothesis generating or thought provoking, that the neoadjuvant immunotherapy works and the PATH-CR also translates into maybe in long-term success also. So this is probably first time we are listening to almost 70%, 80% patients showing response. So this looks a good beginning. In fact, may not be the true result with the larger pool, but at least very good insight, in fact, where we feel that this could be a way to treat them.
True that, sir. True that, sir.
Sorry to interrupt. There is a question in the chat box regarding the mutation.
So no, so if you have a mutation, this won't work, sir. I mean, so I think we have come to that. If it is EGF or ALK positive, it will not really work, sir. So the next one is Empower 010, which basically says it was an interim analysis of the overall survival, which was post-surgery. This was the study. After patient has received 1 to 4 cycles of chemotherapy, you crossed over to atezo versus none. A hierarchical study design, DFS in PDL1 TC from stage more than 1 first, then all randomized stage 2 to 3A, and then DFS in ITT population. And if this is positive, then OS in ITT. So we are right now, this is the interim analysis of the overall survival. Complicated study design actually. I will go not leave to the DFS versus the OS right now. This was the DFS results. So basically based on this result, atezolizumab was approved as the adjuvant therapy in people more than PDL1 1%. And the major and it was not found to be effective in less than 1%. Again, if you see, this is the approval status, and the majority of the benefit was basically, if you see here, was obtained with PDL1 more than 50%. So did you change your practice based on these results? Did you start giving adjuvant atezolizumab, or we are still saying that we don't have many patients? Let's say, Dr. Aditi, if you have a patient tomorrow morning, PDL1 5%, will you give this adjuvant atezolizumab?
So of course, so that's a really difficult question. I will consider. I will tell him the pros and cons.
I am thinking about the prize, you know, which Azharuddin has won. I also may get a prize for the next one. So you are not sure, Dr. Aditi.
Yeah, still I would. I think I'd need more data to advise my patient to go for atezolizumab.
What data do you need, Dr. Aditi?
OS
data? Okay, we will show you OS data also. We will show you OS data. Dr. Tarachan?
Sir, I think we should wait for the OS data first thing. And the data in support of atezolizumab is in the PDL1 positive patients. And again, this has not been tested for the EGFR also, where we have the ADAURA trial also. So there are some
in ADAURA trial, you will not. In ADAURA trial, you will not give. If you somebody is EGFR mutant, you will not give atezolizumab. You may or may not give adjuvant osimertinib. That we will keep on discussing. That's a separate issue altogether.
Yes, sir.
So this was Dr. Aditi, the overall survival of PDL1 more than 32%. At 32.8 follow-up, you had a HR of 0.77. At 46 follow-up, you have 82.1 versus 78.9. So at least at 3 years, these many people are living. So HR is 0.71. Happy or not? I knew somebody will say, "Oh, are you convinced now?" You need some more
yes,
data.
I would give the option. I would give.
Okay. There is an option of giving adjuvant immunotherapy. I think the only issue is, should be given to all the people or only with a stage 3 disease or with a higher PDL?
1 minute, sir. What is happening in the last? Why those curves are mating each other?
No, not really. Actually, if you can see over here,
around you see the after the 60 months. Then why these are the paragraphs are merging? Why these graphs are merging?
I'll show you where the curves actually match. So that is why I was thinking. So this was the chemotherapy trial in IALT trial, where the curves actually after 4.6 years to 7.5. This is where you actually say the curves are matching. After 4 to 7 years, the HR of dying, it was higher in a patient who had received chemotherapy. So this is where I would suggest that the curves are matching. But in the other thing, I think the curves are actually separating, and there's a good, see, if you look also over here, you had a HR of 0.77 and now you have HR of 0.71. So probably immunotherapy, as we all know, the tail of the tail is actually working over there. Let's complicate it further, as you said, and this is the most interesting thing. It was PDL more than 1%, but the majority of the benefit in PDL1 was derived by PDL more than 50%. If you look at TC1 to 49%, your HR is actually 0.95. So that is what I showed Dr. Aditi, that, if you have a NSCLC resected and if you have a PDL1 5%, I will not rather actually give adjuvant immunotherapy, because the majority of the benefit is being derived by more than 50%.
More than 50%.
Since we are just running short of time, I'll complicate it even further. So just see this graph. We are all concerned. This is, this is what actually tells that yes, more than 50% you will have this thing. Let's even complicate further. There was another trial basically of the pembrolizumab in the adjuvant setting, and in this setting, absolutely different results. People of PDL1 more than 50% did not derive benefit. People of PDL1 less than 1% derived benefit. So whether it is because of the statistical design or just by chance, we don't know. So this is exactly my feeling right now. I actually want to bang my head, but this is just the evidence. This was a summary slide by the discussant. I think there's enough evidence to use immunotherapy in stage 2, stage 3. I will use in stage 3. I think subgroup analysis is subgroup analysis. Let's not torture the data too much. Minimal residual disease may be a key to build de-escalation trials, and that is what we think it will come out to be. The final reaction basically was the Canicumab study, and my first reaction on that was APCON. I mean, Atizo malum tha, Pembro malum tha, Nivo malum tha, Dogalo malum tha, abhi Canicumab kaun aa gaya hai. So this was Canicumab study, which Kripa very nicely presented, and this had shown something. This was a trial design. So after chemotherapy, you take Canicumab and placebo, and this was the DFS-OS. So just a one-line answer, Dr. Tarachand, yes or no?
Sorry, sir.
Sir, yes or no? Canicumab, yes or no?
No, sir.
No. Dr. Aditi?
No, sir.
Ankur has already gone. Ankur doesn't want to even listen to Ankur is there. Okay, yes, Ankur, yes or no?
No, sir, not at all, sir.
Okay, Dr. Lalit Mohan also said no. Don't bother me over there. Okay. So I think my conclusion is let's pack up and leave. Canicumab is not really a very good study right now. So just to conclude, immunotherapy is making rapid strides in the adjuvant and neoadjuvant settings. The secondary endpoint subgroup analysis are hypothesis generating, not practice generating. OS data will confirm the utility of immunotherapy in the adjuvant and the neoadjuvant setting. Are we going to cure lung cancer? That is where I'm going to come to, and hopefully we are going to cure lung cancer. So with this, thank you. It was a difficult 25 minutes, something which you don't see very often. But as usual, it was fun discussing with all of you. Thank you.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you, panelists, and thank you, Dr. Ullas. It's always a pleasure listening to you. So now